Assalamu alaikum. Today's our topic is erythropoiesis. Let's start the lecture. The objectives of this topic in which we'll read the blood cells, the erythropoietin, which will lead to erythropoiesis, the hemoglobin and its structure, its metabolism, red cell membranes, and the assessments of the erythropoiesis. Okay, the blood cells, these circulating cells, which forms the pluripotent stem cells in the marrow, divided into main types: the red blood cells, the white blood cells, and the platelets. The most numerous red blood cells, which are specialized for carrying the oxygen from the lungs to the tissue of the carbon dioxide in a reverse direction. Having a 4-month lifespan, that is also called 120 days, whereas the smallest cells, the platelets involved in the hemostasis, circulate for 10 days only. The white cells are made up of 4 types of phagocytes: neutrophils, eosinophils, basophils, monocytes, and lymphocytes. White cells have a wide range of lifespan. The completely pink staining mature erythrocyte results, which is a non-nucleated nucleated biconcave disc. One pronormoblast usually gives rise to 16 mature red cells. The nucleated red cells, normoblasts, are not present in normal human peripheral blood. They appear in the blood if erythropoiesis is occurring outside the marrow. Look at this table. In this table, red blood cells, each and every cell is elaborated and its size, its lifespan, the number, the functions are enumerated over here. So let's start with the red blood cells. Red blood cells having a diameter of 6 to 8 micrometer and their lifespan is 120 days, that is 4 months, and the number, which is in male 4.5 to 6.5 multiplied by 10 is by 12 per liter, and in female 3.9 to 5.6 multiplied by 10 is by 12 per liter function, and the functions included oxygen and carbon dioxide transport. कार्बन डाइऑक्साइड को बॉडी से निकलवाना और ऑक्सीजन को हमारे टिश्यूज तक पहुंचाना. Here comes the platelets, in which their size is 0.5 to 3.0 in diameter, micrometer, sorry. Lifespans in which for 10 days they live, the number 140 to 400, and the hemostasis function. Then comes the phagocytosis, the neutrophils, 12 to 15 in the micrometers, 6 to 10 hours they will live. The number is 1.8 to 7.5 multiplied by 10 is per 9 per liter. Perform the function of protection from bacteria and the fungi. The monocytes having the diameter of 12 to 20, lifespan of 20 to 40 hours, 0.2 to 0.8 multiplied by 10 is per 9 liter their number, and protection from the bacteria and fungi. Eosinophils, 12 to 15 micrometer, and they live for many days. 0.04 to 0.44 multiplied by 10 to 9 per liter, and the protection against the parasites. Eosinophils are basically involved in allergic reactions. Basophils, that is having a diameter of 12 to 15 micrometer and live for days, and 0.01 to 0.1 multiplied by 10 to 9 in number per liter. Lymphocytes, that are in diameter in 7 to 9 in the resting condition, and if they are active they are larger and having the diameter of 12 to 20. Lives for weeks or years as memory piece of the T cells, in the number of 1.5 to 3.5 multiplied by 10 to the 9 per liters. And B cells included immunoglobulin synthesis, and T cells, which involves in the protection against the viruses and the immune functions. Erythropoiesis का function process क्या है? Approximately 10 to the power 12 new erythrocyte cells each day by the complex and the finely regulated process of erythropoiesis that passes from the stem cells to the progenitor cells and the colony-forming units in the granulocytes and the monocytes and the megakaryocytes and breast-forming units and the erythrocyte to the first recognizable erythrocyte precursor in the bone marrow, the pronormoblast. The process occurs in the erythroid niche, in which about 30 erythroid cells at various stages of development surround the central macrophages. And the pronormoblast is a large cell with a dark blue cytoplasm, if you'll see under stain, a central nucleus with the nucleoli and the slightly clumped chromatin. So what is erythropoietin? A hormone which regulates the erythropoiesis. Erythropoietin is a heavily glycosylated polypeptide. 90% of the hormones is produced in the peritubular interstitial cells of the kidney and 10% of the livers and elsewhere. There are no performed stores in the stimulus to the erythropoietin production in the oxygen tension in the tissues of the kidney. Erythropoietin production therefore increases in anemia and also when hemoglobin is unable to give up oxygen. Erythropoietin stimulates the erythropoiesis by increasing the number of the progenitor cells committed to erythropoiesis. So whenever there is low level of oxygen, erythropoietin will produce. The portion of the erythroid cells in the marrow increases, and in the chronic state, there is anatomical expansion of the erythropoiesis in the fatty marrow and sometimes in the extramedullary sites. Increased oxygen supply to the tissue reduces the erythropoietin drive. Plasma erythropoietin levels can be valuable in clinical diagnosis, and they are high in anemias unless this is due to renal failure and if a tumor secreting cells. And let's discuss this diagram. If you'll see the erythropoietin, look at this. Oxygen sensors will sense that there is some hypoxic condition and oxygen is not delivered well. The peritubular interstitial cell of the outer cells will produce erythropoietin, and erythropoietin will produce the cells will mature them to the pronormoblast. Like the stem cells are then converted in the early BFU, the late BFU, the colony forming unit erythrocytes in the pronormoblast that will lead to the formation of the reticulocytes, and after that circulating red blood cells. Then comes the hemoglobin. What hemoglobin is? Heme and globin. Main function of the red blood cells to carry the oxygen of the tissue in the return of the carbon dioxide from the tissues. अच्छा, अब हमारे पास अगर हम हीमोग्लोबिन का स्ट्रक्चर डिस्कस करें जो कि मैंने आपको क्लास में पढ़ाया भी हुआ है कि हीम एक पूरा वो उसका स्ट्रक्चर उसके साथ ग्लोबिन जो है वो अटैच होती है. Each molecules of the normal adult hemoglobin A, the dominant hemoglobin that is mostly present in after the age of 3 to 6 months consists of 4 polypeptide chains, 2 alpha, 2 beta with one own heme group. Normal adult blood also contains small quantities of two other hemoglobins, that is hemoglobin F and hemoglobin A2. There are gamma chains in the hemoglobin F and A2 hemoglobin gamma chains, delta chains are present. Normally percentage 1.5 to 3.2 hemoglobin A2, hemoglobin F that is present in 0.5 to 0.8, and the hemoglobin A that is present into 96 to 98. The structure of the heme. If you'll see there's a globin molecule and the iron is attached. Heme synthesis occurs largely in the mitochondria by a series of biochemical reactions. Pyridoxal phosphate, that is vitamin B6, is a coenzyme for this reaction. And protoporphyrin combines with the iron to form the heme. And the heme will form. And hemoglobin will be formed. The reaction with the globin. Red blood cells in the systemic arterial blood carries the oxygen from the lung to the tissue and return in the venous blood to the carbon dioxide. Alpha 1 chain, beta 1 chain, alpha 2, beta 2 contacts stabilize the molecules and oxygen is unloaded. The beta chain are pulled apart. अब they are describing how oxygen is carried. Oxygen is unloaded by the beta chain and then pulled apart, permitting the entry of the metabolites 2 to 3 diphosphoglycerate, resulting in a lower affinity of the molecules of the for oxygen. This movement is responsible for the sigmoid form of hemoglobin oxygen dissociation curve. Methemoglobinemia is a clinical state in which circulating hemoglobin is present with the iron in oxidized form Fe3 positive instead of Fe2 positive. It may arise because of the hereditary deficiency of methemoglobin reductase deficiency or inheritance of structurally abnormal hemoglobin. So what red blood cells are, I have told you before कि इनका क्या diameter होता है normally. कैरी करते हैं ये ऑक्सीजन को सिंगल जर्नी में कोई तकरीबन अराउंड द बॉडी इट टेक्स 20 seconds and its total journey throughout its 120 days lifespan. The cell is flexible biconcave and with an ability to generate energy as adenophosphate by the anaerobic glycolytic pathways. So the red cells metabolism is done through the Embden-Meyerhof pathway and hexose monophosphate shunt. Its membrane consists of the lipid bilayer, integral membrane proteins, the membrane skeleton, that is 50% of the membrane protein, 20% phospholipid, and 20% cholesterol. The membrane skeleton is formed by the structural proteins alpha and beta, spectrin and chirin and protein. इतनी डिटेल आपको वैसे नहीं चाहिए, लेकिन फिर भी हम इसको एक ओवरव्यू दे रहे हैं. स्ट्रक्चर में अगर आप देखेंगे, आपको ग्लाइकोफॉरिन ए नजर आ रहा है, साथ एनकाइरिन नीचे से जुड़ा हुआ है, अल्फा चेन नजर आ रही है नीचे से, बीटा स्पेक्ट्रम नजर आ रहा है आपको. ये दोनों चेन्स जो हैं वो जुड़ी हुई हैं ग्लाइकोफोरिन सी के साथ. अब इनका हॉरिजॉन्टल इंटरेक्शन अलग है और वर्टिकल इंटरेक्शन अलग है. अगर आप देखें इसका सेल मेम्ब्रेन के साथ. अब नॉर्मेलिटीज में हमारे पास एनीमिया आ जाता है, जिसमें डेफिशिएंसी ऑफ द हीमोग्लोबिन हो जाता है. एज और सेक्स के लिहाज से इन दोनों का अलग कंस्टिट्यूशन है इंसान की बॉडी के अंदर. Presence or absence of clinical features can speed someone's anemia or uncertain severity, age, hemoglobin, oxygen dissociation curve. The most useful classification is that based on the stem cell indices. If you'll see, normoblast is of this type. The whole jolly body looks like this. Basophilic tipping looks like this. Cytoplastic granules look like that. Malarial parasites are this kind of, reticulocytes are these, and Heinz bodies are these. आपको ये differentiation पता होनी चाहिए. आपकी erythropoiesis हर दफा इतनी efficient नहीं रहती है. 10 to 15% of the developing erythroblast die within the marrow without producing mature cells. This is termed ineffective erythropoiesis, and it is substantially increased in the number of chronic anemias. Serum unconjugated bilirubin and lactate dehydrogenase are usually raised, but ineffective erythropoiesis is marked. The reticulocyte count is low in relation to the degree of anemia and to the proportion of the erythroblast in the marrow. So erythropoiesis is being assessed. The total erythropoiesis and the amount of erythropoiesis is effective in producing circulatory red blood cells and can be assessed by examining the bone marrow. Total erythropoiesis is assessed from the marrow cellularity and the myeloid ratio. The raise in proportion to the degree of anemia with the erythropoiesis is effective, but in low when there is ineffective erythropoiesis and abnormality preventing normal marrow response. So इसके साथ ही हमारा erythropoiesis का topic जो है वो खत्म होता है. Erythropoiesis क्या है? If you revise it, तो ये है हमारा red blood cells बनने का process. अगर आप hematopoiesis या hemopoiesis को अगर आप देखें, उसमें आपको पता लगता है कि तमाम के तमाम सेल्स किस तरह से डिफरेंशिएट हो रहे हैं. उसी के अंदर हमने रेड ब्लड सेल्स का बात कर लिया था. उसी बातों को फॉलो करते हुए हमने बताया कि एरिथ्रोपोइसिस कैसे होता है और कौन कौन सी चीजें इसमें हेल्प आउट करती है.